with us. Thanks, Carol, for agreeing to do this. Carol's an affiliate of the SETI Institute, that's right, isn't it? As of quite recently. So that's very exciting. She has a really exciting book, The Quest for a Universal Theory of Life, Searching for Life as We Don't Know It. And Chris and I have both been really enjoying that book, so much recommended. And okay, without further ado, I'll pass over to Carol. Please go ahead.
Okay, let me get my slides up. Okay, so what I want to talk about today is really the materials that's in chapter 8 of my book, and also it was published just before. Oh, that's weird. There's a typo in the, ignore the little e, that's insignificant. I changed the title of the talk to fit the title I gave you guys, and I obviously didn't double check it. But anyhow, it's in chapter 8 of my book, and also in a paper that came out in the journal Astrobiology before my book came out, which I'll give you those references at the end. The paper was called Moving Beyond Definitions in the Search for Extraterrestrial Life. So what I'm going to do is, let me just say a few words about the background of this. So I started out, it all started way back in 2001 with a paper that Chris Chiba and I wrote on defining life. And we argued that it was a mistake to try to define life. And what happened was that as I deepened that discussion, I moved away from, Chris Chiba and I originally wrote the paper jointly, but I moved away from that and wrote some stuff in philosophy, basically more philosophical and logical. And then people kept saying, well, what are you going to replace it with? I mean, if we don't have a definition, how are you going to recognize it when you see it? So that was a big challenge for me. In a sense, it was the inspiration for the book. It's not an accident. Chapter 8 is, there are 9 chapters, and chapter 8 is about that. And so what I'm going to do in this talk is talk about how do you search for life without a definition of life? How can you recognize it if you don't have necessary and sufficient conditions for life? So that's basically the orientation of this talk. And let me start by just pointing out there are really 3 different approaches that astrobiologists have suggested for searching for extraterrestrial life. There's the defining life approach, where you come up with a definition of life, and usually nobody agrees about them. There are many definitions out there. Still a very popular approach. You still see it in astrobiology roadmaps. You still see it in, although it's becoming less popular in astrobiology with the new concept of agnostic criteria for searching for life, which I'm much more sympathetic with, and we can talk about that a little at the end. But the defining life approach is still extremely popular. So that's one approach, and it's an approach that you can see its appeal. Why? Because if you have necessary and sufficient conditions for life, then you're going to be in a much better position to recognize life if the definition's good, because it will tell you whether or not something's alive. It's either thumbs up or thumbs down. It either meets those conditions or it doesn't. But as I've argued, we're in no position really to come up with necessary and sufficient conditions for life broadly construed, as opposed to the life that's familiar life on Earth. And in fact, we don't even know whether or not our popular definitions of life, which by the way, many of them are rivals. They disagree with each other. They're conflicting. That's another big problem. People will fight to the death, literally, metaphorical death, over whether life should be defined metabolically or life should be defined in terms of evolutionary or genetic characteristics. And they don't seem to agree. I mean, you look at Richard Dawkins, he says it's genetics, it's evolutionary genetics. And the metabolic people say, no, it's metabolism. So there's this enormous controversy over what's the best definition of life. And as I'm going to talk about towards the end, when we look at a case study, NASA has tended to go with one definition or another. They went with a metabolic definition for the Viking lander missions, and then they switched to a chemical Darwinian definition in the 90s no accident, I think, that it coincided with a lot of work that was done in molecular biology then, and Dawkins' book was out at that time. But these are conflicting definitions of life. So you're liable to miss something if you're wrong about your definition. So the other strategy people use is they talk about a preliminary universal theory of life. And that's kind of implicit in contemporary practice. People say, oh, I'm not going to use a definition, but these are the conditions that I'm going to use to look for life on Mars or on Europa. And you see this, particularly Steve Benner likes to talk about theory definitions of life. And he and I have gone, shall we say, toe to toe on this topic because his theory definitions of life really are definitions of life. And he doesn't see a difference between a theory and a definition. And in my book, I argue, as those of you who read it, there's a really substantial difference. If you actually look at the nature of scientific theories, they don't provide you with definitions and they can't be summarized in definitions. So that's a problematic account. So what do you do? Well, my proposal that I'm going to develop in this talk is you search for potentially biological anomalies using tentative criteria. I'm putting the tentative criteria aside. I'll talk about that towards the end. But right now, the first bulk of the talk is going to be on why it's potentially biological anomalies that you ought to be looking for. And potential biological anomalies won't satisfy your definition of life because they are things that are going to challenge it. And so that's really important to keep in mind, that an anomaly is something that violates our preconceptions. And I want to look for not just any old anomalies, I want to look for those that are potentially biological. So the question is, how do you do that? But the first question is it really true that potentially biological anomalies have driven discoveries in biology? And in my new book that I'm planning, I'm going to argue they've driven discoveries, anomalies, period, have driven discoveries in all of science. And it's something that's been pretty much ignored, except by Kuhn, and Kuhn's account is inadequate. So how can we recognize truly novel forms of life, examples of life as we don't know it, if we don't have a definition or universal theory of life? This is a challenge I was constantly having thrown at me when I wrote my papers in the early part of the 21st century on why defining life was a mistake. It's not at all obvious that we'll recognize it when we see it. This is something that's constantly said by biologists. Yeah, I can't define it, but I'll know it when I see it. Well, that's just not true. You'll recognize it if it's enough like our form of life. You're going with weaker conditions and to recognize it if we see it, but you're going to have some conditions in mind. So I don't think if it's very different from our form of life, if it's truly novel, it's not the case. You're going to recognize it if you see it, especially if it's microbial. Why? Because if it's microbial, you're going to have to use instruments to detect it, particularly on another world. You're not going to see it with the naked eye. It's going to be small. You're going to need the equivalent of microscopes or the equivalent of cultivation or the equivalent of molecular biology techniques. And those are all designed, particularly the latter two, for microbe that are like our kinds of microbes. You can't cultivate microbes that are really different using a nutrient solution that is used by our form of life, because they won't be able to metabolize it. Similarly, molecular biology techniques wouldn't recognize a form of microbial life that was very different than ours, because most of these techniques require some kind of hybridization with the DNA of the organism. So there are really two strategies for searching for extraterrestrial life. You can search under the street life for life, like searching for keys. You lose your keys and you search for your keys under the street light. So I call this the searching under the street light for life that's like ours. You design equipment and methods for detecting specific kinds of acceptable deviations from familiar life. So this is, I think, up until fairly recently, I think even with the agnostic criteria for searching for life, it's used in that case too. So I think this is a very common method for searching for extraterrestrial life. And I think the agnostic biosignatures approach is very much like this too. And I'm going to suggest something that pushes beyond that without being too critical of the agnostic biosignature, because they've got something right. And then they're searching for anomalies. These are physical-chemical conditions that resemble familiar life in provocative ways and yet also differ from it in baffling ways. Because of this, any definition of life will classify them as non-living. So the idea here is you can't look for life ignoring the features of familiar life that we know about. You have to start from what you know, but you can, and this is going to be the bulk of the talk, you can use the features that we know about in such a way that you can push beyond them, that you don't have them completely blinding you to alternative possibilities. And that's what my tentative criteria that I'll talk about shortly are going to attempt to do. So I want to talk a little about Kuhn. I want to first say I do not agree with most of what Kuhn said, but I think he had a deep insight in his emphasis of anomalies and paradigms. So according to Kuhn, and I know there are scientists here, so if you were all philosophers, I wouldn't be talking about this as much, but I understand I have an interdisciplinary audience. So according to Kuhn, what he calls normal science research, is which mostly goes on in scientific laboratories and field work, is conducted within the confines of paradigms, which include not only theories, but methods, instrumentation, classical experiments, sanctioned text, and most importantly for our purposes, subsidiary assumptions. This is really important. Subsidiary assumptions are assumptions that are kind of just assumed. We'll look at some examples in a minute. They're not necessarily logical consequences of a well-established general theory. They're just things that you assume about a particular domain of investigation. And I think it's really important to, and Kuhn never emphasized the latter, but I'm emphasizing the latter because I think they're driving a lot of our expectations for what we're looking for when we go and look for extraterrestrial life or any other sort of phenomena that is different than familiar Earth life, familiar Earth phenomena. So paradigms are both friend and foe. They are invaluable tools for scientific research. They facilitate the construction of hypotheses, design of experiments, and interpretation of results. You need something like a paradigm. Kuhn eventually, of course, Kuhn's concept of paradigm is so amorphous, he eventually split it into exemplars. So I'm not wedded to Kuhn's point. I'm using the paradigm kind of in a metaphorical sense because I think there's a core truth to that. And as I go on, we can talk about this more later. I think it'll be clear what I have in mind. But the other problem with paradigms, and again, I'm thinking particularly of background assumptions you make, but also theoretical assumptions and instrumental assumptions, et cetera, can nevertheless hinder the exploration of nature because they discourage certain avenues of research and bias the ways in which you interpret the data. And we're going to look at some examples of that actually happening in the course in this talk in a minute. So violation of a paradigm are initially seen, this is again Kuhn, as mere puzzles. I think he's right about this. I think, I don't think the concept of paradigm, I think it's too amorphous, but I think he's right that the initial response to a failed prediction or a puzzling, there I said the word, an observation that doesn't quite fit, is it's a puzzle that we can solve within our current theoretical framework given sufficient ingenuity. But under the right circumstances, a puzzle becomes viewed as something more serious, an anomaly in relation to the reigning paradigm. And once something becomes seen as an anomaly, it's seen as a potential threat to the paradigm or the theoretical framework, which is probably a better way to put this. Anomalies, I'm going to argue, pave the way to scientific discoveries, the heart of the book that I'm currently starting to write, because they weaken the grip of a scientific paradigm or theoretical framework upon widely accepted preconceptions about a domain of natural phenomena. And it's that weakening of the grip that allows one to begin to sort of think beyond the box. And that's what you need to do if you're really looking for something like a novel form of life. I'm not going to go into detail here. I'm going to assume everybody knows that we're dealing with Earth life with a single example. All life on Earth descends from a last universal common ancestor, affectionately known as Luca. And we don't know how different life could have been from Luca. Biochemists have shown us that life could have used a different suite of amino acids to construct its proteins, could have used different nucleobases to construct its nucleic acids, could have used different sugars in DNA and RNA, and we could go on. We don't know whether even you could have very different kinds of informational molecules. These kinds of informational molecules have been experimented with in laboratories. So we don't know whether they're viable under the right circumstances, but we know that modestly different forms of both nucleic acids and proteins could have produced life. And so we don't know how many of the characteristics, the molecular characteristics, especially of familiar Earth life, are due to conditions that were present on the early Earth. And might not be present in other worlds. And also with regard to Darwinian evolution, one of the things that's really striking about Darwinian evolution is that people who are big advocates of evolutionary definitions of life is that we are in a Goldilocks zone for evolutionary definitions. The environment of the Earth doesn't change too extremely, too rapidly for Darwinian evolution to work. That is to say, if you had, we know that during extinction events, for example, usually what happens is that the organism doesn't have time to adapt, particularly multicellular eukaryotes. If you look at microbes, they engage in lateral gene transfer, and because of that, they can adapt much more quickly. So you see the most extensive extinction events, like in the in Cretaceous extinctions, in complex multicellular organisms that are sexually reproducing, because it's just too slow. Mutations plus passing it on to the offspring are just too slow. But bacteria and other, Archaea and bacteria can avoid that because they can swap genes. And they do fairly well under extinction events. They're the last things to go out. So if you had a world that had a much more rapidly changing environment, you might have different mechanisms of evolution. Or a world which had a very slow changing of environment, it might actually evolutionary changes in the sense of responses to the rapidity of mutations might actually be detrimental, and it might not evolve in anything like, it might not change anything like Darwinian evolution. So I don't think we can conclude that, that kind of Darwinian evolution that we engage in is universal. And again, what you're looking for is anomalies. And I just want to point out how little we know really about the possibilities for novel life. So I want to give you the famous Kuhnian example. I think it's worth talking about briefly as an example of of how anomalies can drive scientific discovery. So discovery of Uranus is an interesting example. So it turned out that up until 1690, between 1690 and 1780, astronomers had reported a star in the place we now know is occupied by Uranus. They didn't see anything funny about it. It was just another star. Herschel developed a telescope that was better than the previous telescope. And when he trained his telescope on various stellar objects, he noticed that there was something funny about Uranus. It had a disk shape, which was unusual for a star, and it moved among rather than with the stars. And so Herschel concluded that he discovered a comet. Now, Kuhn argues that up until that time, the view was something was either a comet or a star or a planet. There was no, and there were only supposed to be 9 planets. So he could not fit this into any of the existing categories. What happened was that Lexell, about 6 months later, because everybody got excited when Herschel said, hey, this object that we thought was a star really is anomalous. It doesn't fit. And I think it's a comet. What Lexell did is he analyzed the orbit just 6 months later and said, hey, it's not cometary. It's not a cometary orbit. It's a planetary orbit. And we discovered there was another planet, Uranus, in our universe. So the view that there are no planets beyond Saturn was rejected, and we discovered that there's another planet. Now, what's interesting about this is that within a very short time after the discovery of Uranus, astronomers suddenly saw many planetoids. Most of them were probably in the asteroid belt, but many planetoids, non-cometary orbits that they hadn't recognized before. So using the same inferior telescopes, they could recognize Uranus was actually had an aberrant orbit, and there were other objects that had aberrant orbit, whereas before they totally ignored it. So here's an example of how you can be blinded by a paradigm and or a theoretical framework and not even recognize when it's standing in front of you that something's anomalous. And of course, there are the really fun psychological experiments where they introduce a gorilla into a room and people don't notice it's a gorilla. So it's that kind of phenomenon. I want to talk about now some cases that Kuhn doesn't talk about that I think are important for thinking about the role of anomalies in scientific discovery. So one of the famous cases, and I have a lovely quote to show you in a minute, is the discovery of catalytic RNA or ribozymes. Basically, the discovery challenged the central dogma of molecular biology, as stated by Crick, holding that there is a rigid functional division between nucleic acids and proteins. Proteins supply the catalytic material for life. Nucleic acids cannot play the role of biocatalysts. So the view was that you have informational molecules, and RNA is an informational molecule that translates the information from DNA, and of course is used to transcribe it in ribosomes into protein. And that, you have to have protein catalysts to do all of these transitional processes like transcription and translation. So there was this kind of the chicken and egg problem, but this was the central dogma of biology that you had to have, only proteins could be catalysts. So Tom Cech and his team, of course, he received the Nobel Prize for it. Sidney Altman shared the Nobel Prize for the discovery of ribozymes, but I'm going to talk about Cech's work because he was at CU Boulder, and I happen to know the man and have heard, he's told me about how this happened. So Cech and his team discovered catalytic activity, splicing, in a preparation of RNA from which they thought they had removed all protein. So the basic idea here is that that when you have RNA takes out chunks. It basically takes out chunks of when it's being transcribed, and then it has to splice together the ends. So they wondered, how does this happen? And so the theory at the time was there's some protein that does this. And they were trying to identify the protein that does the splicing. And they had a sample of RNA from which they thought they'd removed all the protein, and yet they kept getting splicing. They tried every way they could think of to remove proteins from it, and still they got splicing. It didn't occur to them that the splicing might have been from the RNA itself. They kept thinking they just had failed to be able to identify the protein. It took them a year before they finally accepted the idea that RNA might be catalyzing itself. But the wonderful quote, here's a quote from Tom Cech's Nobel lecture, really brings out the fact that this was an anomaly, and the theoretical framework, the central dogma, which they were working under, was blinding them to what was going on. He says, and this is a quote, "Our first hypothesis was that the splicing activity was a protein tightly bound, perhaps even covalently bound." That's a very implausible assumption, but they were more willing to go with that assumption than the assumption that the central dogma was false and that the RNA was catalyzing the splicing. "Our first hypothesis was splicing activity is a protein tightly bound, perhaps even covalently bound to the pre-RNA isolated from the tetrahymena nuclei. This would have to be a very unusual protein RNA complex to survive the multiple forms of abuse to which they had subjected it." So they had heated it up to high temperatures. They had used various other kinds of chemicals to denature proteins, and they could not get the splicing to cease. That we took this hypothesis seriously says Check, provides an indication how deeply we are steeped in the prevailing wisdom that only proteins were capable of highly efficient and specific biological catalysis. So this is a really nice, I think, illustration that's biological as opposed to the Uranus illustration of the kind of grip that assumptions that one has. They don't necessarily be assumptions from a broad general theory of life. This was called the Central Dogma for a reason. It was a dogma, but it had such a grip on the imagination of Check and his team that the last thing they were willing to consider, and it took over a year for them to do this, was that maybe the problem was that the RNA was actually doing the splicing. It was catalytic, and basically that the central dogma was false. So that's a nice example. Discovery of mimiviruses, these are giant viruses, violated assumptions about the size of viruses. Basically, they were initially classified as bacteria on the basis of their very large size and their gram-positive staining. So here's just basically preconceptions that biologists have accepted that, if it stains gram-positive, then it must be a bacterium, because bacteria stain gram-positive and negative. And if it's large, these were large, they weren't gigantic, but they were larger than viruses. And the view was that viruses don't need to be big because they don't have to have all of that genetic material, inside them. They basically parasitize other cells and use their genetic machinery to make more of them. So it was viewed that viruses are always small. They don't need to have that genetic material for translation and transcription that you do in bacteria. And that's why bacteria are big. It wasn't until 2003, 10 years, that they were finally reclassified as viruses. It was just assumed on the basis of their large size and their gram-positive staining that they were actually bacteria. And an even better example is Pandora virus, which is larger than parasitic bacteria. It's bigger than some bacteria and visible under a light microscope. And they too were viewed as a weird kind of bacteria until they were discovered. Actually, what happened with Pandora viruses is kind of fun. I'm sort of misstating that. Once you discovered that viruses could be bigger, people began to see large viruses like the Pandora virus everywhere. Pandora virus had not been recognized or even discussed as being not a virus or a bacteria. It was just pretty much ignored until you discovered these larger viruses, and then people began to look for them. And so that's kind of like the sudden discovery of many planetoids with planetary orbits after the discovery that Uranus was a planet and there were planets beyond Saturn. So another good example is discovery of the Archaea. So initially, by Carl Woese and Sidney Fox, the kingdoms of life were the dominant way of categorizing life on Earth into 5 kingdoms. There are the Monera, which included what we now know are bacteria and archaea, all these single-celled prokaryotes. And then there were the protists, the fungi, the animal, and the plant. But they were all considered to be equal kingdoms. They were all distinctive. And then with Carl Woese's genetic investigations, where he analyzed the genetic, he basically sequenced the genes of all of these 5 kingdoms, and he discovered something astonishing, that the eukarya were actually, so he discovered something astonishing. What had been considered Monera actually split into 2 different categories, such that these categories were more different than each other in their genetic machinery and the lipids in their cell membranes than they were from these things, the eukarya, the protists, fungi, animalia, and plants. And so basically what happened here was that you had 3 domains superseding the 5 kingdoms, and the 5 kingdoms became 6 kingdoms. But what's really interesting is these organisms, on the basis of their morphology, their prokaryotic cell structure, have been classified the same. When you went and looked at their sequenced their genes, you discovered that the archaea and bacteria were more different from each other than they were from the eukarya. The eukarya and the archaea were more similar in their genetic machinery than they were to the bacteria. And the archaea and the bacteria and the archaea were more different from all of these in the lipids in the cell membranes. Pretty astonishing, but an example again of, and as many of you know, Tom Cech received horrible, I mean, he was, for many years, he was given a very hard time. And he became very, as some of my colleagues have told me, paranoid, because he was so heavily criticized when he initially released this proposal of switching from 3 domains to 6 kingdoms. He was called, people just rejected it. They said, this is ridiculous, etc. Here's a lovely quote before, not just about the time that he was doing his discoveries. The fact that this is from a textbook on thermophilic microorganisms and life at high temperatures. It was used as a textbook in the late 1970s. The fact that sulfolobus and thermoplasma have similar lipids is of interest. Remember I said that the lipids are really different in the archaea than they are from the bacteria. The fact that sulfolobus, and thermoplasma have similar lipids is of interest, but almost certainly this can be explained by convergent evolutions. So they're treating it as a puzzle, what Kuhn would call a puzzle. They're saying, well, the lipids are really different here, but we can explain this within our framework and our current classification systems. It's convergent evolution. This hypothesis is straightened by the fact that Heliobacterium, another quite different organism, has similar lipids to those two acidophilic thermophiles. All three are archaea. So this is another example of how our preconceptions can blind us to a really important discovery. These organisms are anomalies, but they weren't recognized as such until basically Carl Woese went in there and did a genetic analysis, and all three of them are really different than bacteria, both in the lipids in their membranes and also their genetic machinery. Their genetic machinery is more like that of the eukaryotes. So the role of anomalies in scientific revolutions, all of these cases involve phenomena that, while present all along, were unrecognized for what they represented because they conflicted with the assumptions or categories provided by a reigning scientific paradigm. And notice these are not big sweeping theories like general relativity. These are basically, assumptions that are just kind of embedded in this theoretical structure, things like the Central Dogma, or views about viruses, basically having to be much smaller than bacteria because they don't have any need for ribosomes and the other kinds of machinery that you find in cells that are, ribosomes would be an example, but there are other kinds of machinery that you find in cells that take up space. They were either not detected because they weren't looked for, or they were detected but misidentified because they violated prevailing scientific assumptions. That, I think, is exactly what happens when an anomaly is viewed as just a puzzle that can be solved by convergent evolution or some other explanation. So my solution: explicitly search for potentially biological anomalies using tentative versus defining criteria for life. This is my proposal to the challenge. Well, how are we going to recognize it when we see it? It's not as satisfying as a definitional approach, because it turns out that all you can do on my account is that you recognize that something is worthy of further scientific exploration. Potentially biological anomalies are physical systems that defy classification as life or non-life because they violate our Earth-centric preconceptions about life. Tentative versus defining criteria for life include both allegedly fundamental and non-fundamental, universal and contingent characteristics of familiar Earth life, which are deployed jointly for the purpose of detecting not life, but potentially biological anomalies. And I'm going to, at the very end of the talk a little more about what counts as a tentative criteria, but I want to move on in a minute to an example from astrobiology, where I think we actually have discovered a potentially biological anomaly. But I want to emphasize now that when you identify an anomalous physical chemical system, you haven't decided the category it fits, whether it's life or non-life, but it becomes a focus for further scientific investigation. This contrasts with the definitional and preliminary theory approaches, which are focused on finding life per se. Their goal is to say it's life or not life, and identify cases that do not conform to the set of privileged characteristics and use, privileged or eccentric, I might ask, characteristics and use as non-living, period. And here's something really important, because I struggled with this when I wrote the book. There's no algorithmic decision procedure for detecting anomalies. I was hoping I could do it Bayesianism. I tried, or statistics, but then it suddenly occurred to me, you can't anticipate the unexpected before encountering it. So I finally gave up on trying to have an algorithm for detecting whether something was an anomaly or more further investigation. And you'll see as I get to the end why that's such a hopeless project. So I want to give a quick case study. How am I doing for time?
It is 20 to 6. You've been on for about 35 minutes. Take another.
Oh, good. So I could
basically.
So I'm okay with time then.
Absolutely, yeah.
Okay, good. Then I'm not going to rush through this. So I want to talk about a case study where I think a potentially biological anomaly was encountered but dismissed. And I think that was a mistake. So I'm going to talk about the Viking missions to Mars in the mid-1970s. And by the way, people are beginning to revisit this case because the grip of the sort of theoretical framework upon which they were working has loosened. So I'm going to focus on the Viking labeled release experiment, although all three, there were basically three metabolic experiments. All of them initially produced what were positive results, and then on further investigation, two of them could be more easily reconciled with our then current theoretical framework. The Viking labeled release experiment was the hardest one to reconcile. So let's talk about how that one was handled by NASA and by the PIs, NASA's PIs of that experiment, Levin and Stratt. So Levin and Stratt concluded yes. And let me explain that they had found life. And to this day, Levin still fights about that and claims he found life and he was cheated. I think basically Patricia Stratt has become kind of quiet about it and recognized that no way she's going to convince people. But they both initially concluded yes. So let's talk about that experiment. So the basic idea was they had a nutrient solution labeled with carbon-14, radioactive carbon. And they had tested this nutrient solution on, and this is in the mid-1970s, remember, on bacteria, a wide variety that they could cultivate. At that time, it was not recognized, Norm Pace had not done his work, that we can cultivate less than 95% of microorganisms on Earth. That was pretty astonishing. At that time, people were very naive about microorganisms, and the ones that they could cultivate were the ones they knew about. And they saw that they could come up with this nutrient solution, which was radioactively labeled, which all of them could metabolize at least part of it. So that was a nutrient solution they went to. At that time, it wasn't recognized that there are a lot of bacteria that are not organotropes, that some of them eat hydrogen gas, some of them eat iron. I mean, so there was a lot of ignorance about the composition of microbes on Earth. But at that time, they were testing it on ones they could cultivate. And now we know that was only a small snapshot of the microbes on Earth. So that was their first mistake, but they didn't know it. So it's a good example, again, of preconceptions that people had at that time, and the role of preconceptions in one's reasoning about natural phenomena. So what they did is they took this nutrient sample, they took it to Mars, they had a lander or two landers. It took a bunch of Martian soil and put it in the hopper, and then it wetted it with the nutrient solution, and it waited to see whether or not carbon dioxide gas would be released as the hopefully Martian microbes metabolized the nutrient solution. And they got that. it released carbon dioxide gas that was radioactively labeled. And they had one control experiment that had been planned in advance. And that was to heat the Martian soil to a temperature of 160 degrees for 3 hours, which was a temperature high enough to kill any known bacteria. And when they did that, when they took Martian soil and heated it and wetted it with the nutrient solution, it failed to release carbon dioxide gas. So they were very excited because it did just what they had agreed in advance. If there was microbial life on Mars, would happen. It's clear that they were working with a metabolic concept of life, because that's what they're testing for, is metabolism. And on the basis of the agreement that these two experiments, if they got positive results, would be life, Levin and Stratt concluded they'd found Martian life. In Levin's words, the Viking mission labeled release life detection experiment returned data from Mars fully satisfying the pre-mission, and he talks about it as defining criteria for the detection of microbial metabolism. So he felt NASA was cheating if they didn't go with we discovered life. NASA concluded no on the following really interesting empirical grounds. First of all, they had this very primitive GCMS, and it didn't detect any organic compounds in the Martian soil. What's ironic is later it was shown that GCMS wouldn't have recognized organic compounds in Antarctic soil containing 10 to the 6th bacteria. That's a pretty bad defect. 10 to the 6th bacteria in a small handful of Antarctic soil, and it couldn't have recognized, it could not have detected organic compounds. And now we have discovered organic compounds. But at the time, there were no organic compounds found by the GCMS. And so NASA used that as their main reason for concluding they hadn't found life. It's particularly ironic since we know there are organic compounds on Mars now. We have detected them. And at the time, it was puzzling because we know that meteoritic dust is constantly falling on Mars as it does on Earth, and it contains organic compounds. So there should have been suspicion about why they couldn't find organic compounds, and they could explain that away potentially in terms of basically an antioxidant a very powerful oxidant in the soil of Mars, and that's what they did. Here are some other reasons they gave. The LR response from Mars was thought to be too much too soon. I find this one really very Earth-centric. So the LR response, the release of the radioactively labeled carbon dioxide gas, was fast. It was too fast. It had the same kind of curve as we have on Earth, only it was faster. It was higher than the curve that you find on Earth. So basically, if it didn't metabolize like it does on Earth, it couldn't be life, was their conclusion. They were at this stage, I think it looks like, fishing for ways of saying they hadn't found life. A second injection, and here is some of the most critical evidence that was used, a second injection of LR nutrients. So what they did is they gave an injection. They got the release of the radioactively labeled carbon dioxide gas. They waited a little bit so the hungry Martians, microbes that they thought had released that gas, metabolized the nutrient solution, would be ready to do it again. But when they gave a second injection into that soil sample that had previously released the gas, not only did it fail to reinvigorate the evolution of nutrient gas, which is what happens with microbial containing Earth soils, but also, and this is deeply paradoxical, about 30% of the gas evolved after the first injection was reabsorbed by the soil. And that never happens in microbial containing Earth soil. So here we have what looks to me like definite evidence of an anomaly. How did they treat the anomaly? Well, they were going with a metabolic definition of life, and they even made in some way implicitly that definition more specific in these responses. It had to be just like Earth life, same kind of curve. It couldn't be faster than Earth life, and it had to behave just like Earth life. And so they concluded no, they had not found life. Various abiological candidates for this mysterious Martian oxidant, which was supposed to explain why there was no organic compounds on Mars, has been explored over the years, including hydrogen peroxide, superoxides, exotics, oxidation states of iron, and most recently perchlorate, which was accidentally discovered in the Martian soil. But it turns out perchlorate is actually not a good explanation. Why? Because perchlorate is not particularly reactive unless it reaches a high temperature. And the temperature of the Martian soil in the experiment in the lander was only 10 degrees. It wasn't high enough to make perchlorate reactive. So what they did is they said, well, maybe on the Martian surface, there are various reactions, partly reactions as a result of light and other chemicals that turn perchlorate into a more reactive, stable reactive substance. And so that could explain why you didn't find any organic compounds in the GCMS, and it could explain the results of the LR experiment. But the important thing here is, even in the case of perchlorate, which is now often cited as having settled the issue, if you look at the literature actually on perchlorate, it's really difficult the kind of reactions required to make perchlorate fit the results, explain the results of the LR experiment, are highly speculative and not clearly something that happens on the Martian surface. It's not clear that perchlorate degrades on the Martian surface into a highly reactive substances, which can then act as an oxidant at lower temperatures of the sort that they found actually in the were found in the experiment on the lander. So here's an option that hasn't really been considered. Life's an oxidant, right? They're considering all these exotic oxidants here. The one thing they're not willing to consider is one particular oxidant, life. And I found on the web. It's really fun. It's owl oxidant. It's some band, but I thought it was really appropriate since we're looking for kind of an owl oxidant, a biological oxidant. But that's not what they have considered. Recently, very recently, in the last 5 years, people have gone back and looked and taken more seriously the possibility that actually what was discovered was an oxidant. But people are really worried because they say, well, it didn't dig deep enough and you couldn't find, and life would not survive so close to the surface of Mars, so probably it wasn't life. Again, what do you see here? You see life has to be like our kind of life. There's no assumption that life could have adapted to the Martian surface in such a way that it could have survived, maybe not very far under the surface, in the material that the hopper picked up in the Martian experiment. After all, we know that there are bacteria now that live in radioactive material. They actually can live in radioactive. They can survive living in nuclear power plants. So again, we're basing our decisions here on very Earth-centric assumptions about life. The truth is, neither side of the debate over the labeled release experiment knew what they had found. The results of the labeled release experiment were potentially biologically anomalous. Notice the potentially here. I'm not saying they were biological anomalous. I'm saying they're potentially biological anomalous. Maybe more research would reveal that, A, this isn't a biological anomaly. We can explain it abiologically. The important point here is that what I am suggesting is you look for potentially biological anomalies. You explicitly look for them and treat them then as potentially biological. They could be biotic, they could be abiotic. And that's what didn't happen with regard to the LR experiment. Nobody designed until recently instrument packages to go back and investigate what the character of that mysterious Martian supposedly inorganic oxidant was. I once asked a head of NASA, this was quite a while ago, why they hadn't done that. And he said, well, we're looking for life and we didn't find it. And again, Rather than recognizing they had a potential biological anomaly, their conclusion was they hadn't found life. I think that's the typical response. I think Kuhn gets something right when he says that basically our theoretical framework, what he calls a paradigm, allows us to treat something as a puzzle or as an anomaly. And I think that it makes perfect sense initially to treat it as a puzzle. But as time goes on and you have to come up with many, many explanations, none of which are satisfying, surely it's time to think about it as potentially biologically anomalous. So that basically is the whole basic framework that I have in mind. And I want to just now list some candidates for tentative criteria for searching for potentially biological anomalies, just to emphasize how different this approach is than an approach based on defining criteria. So if you're looking, so tentative criteria are differ from defining criteria in some really important ways. Allegedly fundamental defining characteristics of life, metabolic characteristics versus evolutionary genetic characteristics, aren't viewed as separate. So you fight over which definition you're going to go with, whether you're going to go with a metabolic definition in designing your biological experiments, or you're going to go with a genetic evolutionary or chemical Darwinian definition. In my view, you go with it all. You don't view them as defining, but rather they're universal characteristics of familiar life that might be, in fact, due to contingent features that were basically at the origin of life or inherited from Luca, the common ancestor of all life on Earth. You deploy them together. So you don't sort of commit yourself to having one or the other exclusively. Universal characteristics of life that are not thought to be fundamental to life. You might look for DNA or ribosomes. A lot of people have argued that there's PNA and other possibilities for genetic molecules. So you can look for, DNA or ribosomes or other kinds of characteristics of life as we know it, but you don't take it as truly universal to all life. And here's the most important thing. You look for contingent, non-universal ways in which organisms found in certain environments on Earth modify them, sedimentary structures, reactive gases. But again, you don't view them as conclusive for deciding the case. You might, for example, in an environment, so for example, magnetotactic bacteria are particularly interesting because on Earth, you only find them in certain kinds of boggy environments because they use these little magnetite crystal strings of them to help them keep at the same, orientation and level in a boggy environment. Where they have enough oxygen and food, etc. So you might consider, if you're looking in a boggy environment on another world, looking for looking for tiny magnetite crystals. That's just an example. Or sedimentary structures. Characteristics rare among living systems, but even more rare in non-living systems. I guess I had magnetite crystals down here in the pertinent analogous environment. So you don't find magnetite crystals in those kinds of environments. The only magnetite crystals you find in those kinds of boggy environments are from magnetotactic bacteria, and they have very specific characteristics that are almost geometrically perfect because the bacteria pack magnetite into these little packets, these little biological membrane enclosed packets. So they're almost geometrically perfect, and they are almost chemically pure. So they have certain kinds of characteristics. So you use all of these, and what you look for something that shouldn't be there, something that basically has a number of these characteristics that are provocatively suggestive of life, but also these unanticipated characteristics that are suggestive of non-life. And that's the end of my talk. And here are the two papers in which I developed these ideas. The first paper is the one in astrobiology, and the second is my chapter 8 of my book. Thank you.
Thank you very much, Carol. That was fantastic. I really enjoyed that. Thank you. So we have